So today I want to talk about the phasor approach application and also to FRET analysis, but essentially I will repeat something that Dave says because we were working together on this project, but I will put the accent on the fact of FLIM. And the FLIM essentially is measurement of phasor on 100,000 of a million points. And that gives a possibility to do some sort of statistics and apply artificial intelligence method that otherwise from a single point you cannot apply easily. So, but I want to insist about the fact that was the propagation of error that really makes the difference in application of phasors and so on. So let us start with the, okay, so let's see if I can start. So I will say what is lifetime, which probably doesn't need to be done anymore after Dave's presentation, but I want to set the motivation for FLIM. He talked about motivation for lifetime, but I will talk about motivation for FLIM, why you want to do FLIM. And then some, what is the phasor approach and what are biosensor and eventually future prospects of that. So Dave already talked about what you measure. So you excite the molecule from a ground electronic state to an excited electronic state. There is a very fast decay to the bottom of the excited electronic state. Essentially, every one of those lines represents a vibration, and this is about a picosecond per vibration. So after 2-3 picoseconds, you get to that point, and then you get the fluorescence. And this is the signal that we measure. Okay. And the point of FLIM is to do the measurement of the decay in every single point of an image. So, for example, in this point will be a time t equal to 0, time t equal to t1, t3, and the intensity will change. And this is really the point. So the point is that we have an image. So an image is where if you have 256 by 256 is 600,000 points. If you have 1024 by 1024, will be about 1 million points. So we have 1 million points in the phasor plot, and that makes a difference in the way you do the analysis. So let us see some of the properties of the phasor plot. So Dave says that it's possible to do a frequency domain, which is true, but in the frequency domain, what he didn't say very clearly is that the error on each phasor propagate at the different frequencies in a different way. And this is because the frequencies are like the Fourier transform. And so the Fourier transform are one independent from the other, and that propagate errors in a very different way. So why we want to do FLIM? FLIM is, for example, to measure FRET, Foster resonance image transfer in every point of the image, but we can use FLIM also to intracellular mapping of ion concentration at pH imaging, for example. This is very, very important. We can study biochemical reaction, and I will talk a little bit about that. And for example, in the process of NAD versus NADH, which have different lifetime and different things. And then recently, we have been working a lot about the long lifetime imaging through phosphorescence. For example, measure oxygen concentration of cell or in tissue. This is very important because essentially the metabolism of a cell depends, of course, on the presence of biochemical reaction like NADH and NAD, but also on the oxygen concentration. And so to be able to measure simultaneously both things is very important. For example, the concentration of oxygen can be different in films of bacteria or in the soil, or it is very important for our planet in this moment. Okay. So let us start a little bit about resonance energy transfer that was done rapidly by Dave. So we have a donor which is excited, so that means it is excited, and then transfer energy to an acceptor, and then the acceptor will be excited after with the rate of kT, which is the transfer energy, and eventually can come back. Of course, can come back only if there is the energy of the acceptor is equal to the energy of the donor, and that happens only if you have a homotransfer or something like that. Okay. So, but the important thing is this delay. So the acceptor is not excited at the same time than the donor, but is excited at the later time. And that is what Dave says, that you get a delay, and the delay in this delay gives rise to a phasor to be outside the universal circle. And this is very important because that is a signature that there is a delay. And that signature occurs not only if you have FRET, but also if you have excited series reaction or other kind of things that we studied in my lab. So, and I will not go through Foster derivation, but I want to simply to say that the emission and the absorption have to the emission of the donor and the absorption of the acceptor has to overlap in certain way, otherwise cannot be changed. Okay, so one important thing about measurement of energy transfer is that energy transfer can be measured either by a lifetime or by fluorescence decay, fluorescence intensity. And this is the difference, that the lifetime in the presence of the donor, that will be the lifetime of the donor, is very easy to obtain simply because you do a sample without the acceptor, or essentially, this is very simple. And then you have a sample that has the acceptor, and then the efficiency of energy transfer can be calculated directly by this expression, which is what we use in order to calculate the energy transfer. Of course, the energy transfer can also be calculated and measured in fluorescence. But for example, the fluorescence of the donor only, it essentially has to be done on a cell, which is different than the fluorescence of when you measure the lifetime. And that generally, the cell is different, so we have different background and so on. So there are many terms that are used to correct that. And then when you have in the presence of the acceptor, also you have many other terms that you need to correct. So clearly, that shows that the measurement of energy transfer by lifetime is much easier than done in other ways. Okay, so this is an example, which is a paper by Roger Sien, in which for the chameleon protein, and then the chameleon protein shows the donor, which decreases as the acceptor concentration is increased. And this is the ratio that Dave was talking about. Okay, so let us talk a little bit about why you want to do FLIM. So, for example, you want to do FLIM to identify molecular species. So, for example, that will be the CFP, that will be the YFP, and then you can measure the lifetime components. So, CFP in the presence of YFP, and with FRET, you will get the curvature in the time decay, and you use some sort of either spectral demixing or multi-exponential decay to obtain the amount of FRET that you have. So essentially, in this example, you can see that using spectra or using lifetime, you will get a very similar result. Actually, you have to get very similar result because you are using the same chemical species. Okay, so let us talk a little bit about the challenges of FLIM that are not the same challenge that Dave has in the cuvette, because you have essentially purified substance and so on. So at every pixel of an image, you have the contribution of several fluorescent species, and each one could be multi-exponential. So you have a problem which is very different. And yes, you can purify the monomer and the dimer of HSA and so on, but you cannot do that in a tissue or in a cell because the cell is expressing proteins, whatever they are. But this is not really so. First of all, I want to make clear that there are many peaks. At every pixel, you have a contribution of several fluorescent species. Each one can be multi-exponential. So this is a problem in FLIM. So to make things worse, is that we can only collect light for a limited amount of time. For example, 100 to 200 microseconds. Why is that? Because if you take seconds to collect it for a pixel and you have 1 million pixels, that will make an entire day of measurement. And so clearly that tells us that we can have only a certain number of photons per pixel. And this is important because in all the curves that Dave showed, if you have very low counts, you have also very large noise. And so this is important to understand. So if you have 500 to 1,000 pixels and you want to distinguish between a double exponential from a single exponential, this is almost impossible because the curves superimpose too much. So, and that is where the noise came out to be important because the noise that you have in measuring every pixel in terms of photon is different than the noise that you will have if you measure phasors. And I will show you that. Resolving the decay and each pixel in multiple components involves fitting a function, which is traditionally a complex computational task. And then we say that only if you are an expert and you know already what the lifetime are, it is next to impossible to solve the problem. I don't like that people need to be expert in order to solve a problem, because that means that only few people can do it. You need years of experience and so on. So this is a problem. So you have few photons. You have to be able to distinguish several exponential. And then in order to do that, you do fits like Dave says, but that fit it becomes very difficult to do because essentially either you know very well how to start from a given position, or otherwise you don't do that. One thing that people try to do, and I tried to do, was to analyze, to say, well, in an image, you have only 2 or 3 lifetime, and then you get the abundance of those lifetime pixel by pixel. But of course, this is not true, because you can have a region of pixel, might be 10 pixels or 20 pixels that have different lifetime, and that is a wrong assumption. So the major problem with FLIM is that data analysis and to be an expert in the field, and we don't like that. So there are clearly major issues of FLIM. So it's a rather difficult technique because you have to collect the lifetime at many different points. Fitting is low. Sometimes, the global program I was using was taking 2 or 3 hours, and I cannot wait 2 or 3 hours to get an answer. And the result depends on the initial condition, which means interpretation requires expertise. So those are the major issues with FLIM, and for many years people were unable to use FLIM to do analysis. But today, if you look, for example, in phasor analysis for lifetime, not phasor analysis, because Dave says you get millions of entries in Google, but if you look phasor analysis for lifetime, you get about 60,000 entries, and all of them are using phasor, because that's in the title of the question. And so clearly has become a very popular technique. So the question is how to avoid those problems. Well, the major thing is that I want to have no expertise necessary. And this is what the phasor plot, the phasor analysis will give you. I want to have instantaneous result instead of waiting for hours. And this is also something that the phasor analysis will do. I want to be independent of the initial choice, which is requires no expertise. I want to have fully quantitative result, so maintain the quantitative result that we can obtain with FIT. And we want to have a very simple interface, which everybody can use. And that is really the point. And in order to do that, we need to move to a different space. So really specifically, the space is the space of the Fourier transform. So we need to work with Fourier harmonics. And if you ever have done that, you know that Fourier transform is very easy to do. Essentially, 1 million of Fourier transform can be obtained in less than a second. And so you can do your measurement project in a different space and then move in that space and do the analysis in that space. And that is essentially shown here. So this is a phasor, a spatial gun, and clearly those guys are able to do everything that we cannot do as in our planet. But where is the concept coming from and where is the math of that? So let us start with something I already showed you, that we have an excitation pulse, you have the decay, and you want to measure that decay. So in a FLIM experiment, the sample, so that means the image, is flashed many times by a short duration laser pulse, general femtosecond pulses or something like that. The interval between the excitation flash and the first excited photon is measured, as they say. So you build a histogram of this decay. So let us see what is the transformation that we do. We follow Gregorio Weber, but in a slightly different way. So I don't know if you noticed, Gregorio Weber introduced the G and S coordinate as a cosine and sine transform of the intensity decay, but we normalize the decay by the sum of all the photons, the total intensity. This is needed in order to get the phasor that will have the properties I will talk about. So in some way, that expression already tells you that the component of the phasor are weighted by the intensity, because this is the total intensity of the sample. And that is important because we don't measure a pre-exponential factor, although we can obtain the pre-exponential factor, but we measure a fractional intensity. And this is, every point is represented by fractional intensity. So you can see that very clearly from the expression. So I of t is a decay. So it can contain many exponential or be a single exponential, but we will analyze the case separately. So as Dave said, if the points are single exponentials, they must follow in the so-called semi-circle, which is the point of all the, so if the case is single exponential, it will fall in that circle. So that means that if you are not an expert, that it cannot be here and cannot be there, a single exponential must be on that circle. And that reduces enormously the search for the solution. But that has a consequence. And the consequence is that if you have a single exponential, for example, with length time t1, as she showed, and another single exponential with length time t2, that if you have an experimental point which is in between t1 and t2, that means that there is a combination, a linear combination between tau1 and tau2 that gives the experimental point, and that is the line. So that means that instead of searching every place for tail X time, you are only restricted to tail X time that are along the line of T1, T2. And this is really what makes things very easy, because if you have tau1 and tau2, and I will show you the cases in which you have tau1 and tau2, and then you get this experimental point, and this experimental point is given by the linear combination of tau1 and tau2. And this is fundamental because at the moment you have might be 1 million points, you will search only in the line that goes from tau1 and tau2, instead of searching everywhere. And that search is done, of course, in an easy way by a computer, and I will show you how that works. So, so let us talk about the process we follow. So in the time domain, we have the decay. So we have to solve fitting exponential, which is not an easy thing because you have to know where to start. And then you get the lifetime distribution. And this lifetime distribution, please remember this lifetime distribution, which means the abundance of this lifetime or the other lifetime. So it's the number of pixels that has one lifetime and the other lifetime. In the in the phasor plot, you first transform the decay in the phasor, and then you get the lifetime distribution. And that is a very different process because it does not require fit. Here, we require a fit of the decay in order to obtain the lifetime distribution. Here, we do not require the fit in order to get the lifetime distribution. Okay, so let us make some example. So this is three capillaries, one containing the fluorescein, the other containing rhodamine B, and the other containing the mixture of the two. And then what you have is that will be the fluorescein in the phasor plot, that will be the rhodamine B. Notice that it's a little bit wider than the fluorescein. And then if you trace a line between the fluorescein and the rhodamine and the fluorescein, you get experimental point. And then if you select this value of the lifetime, you paint with a given color, the L. If you trace the other, you paint with a different color, the D. And then at the end, if you select the other one, you paint a mixture. And you see that I have analyzed the sample to say that I have a mixture of two components without ever doing a fit. And this purely graphical. And this is what people like because you don't have to be an expert. I can tell any person to do this kind of analysis. And so this analysis is very simple and very easy. But you will say, well, but I need to know the lifetime of one and the lifetime of two in order to get the sample. Well, this is not true. And this is the other important thing about the phasor plot. Okay, let us take a sample, which is a Paxillin EGFP, a cell expressing Paxillin GFP, and you see the cell, and then you see essentially the cell. So this is the phasor plot, and you can see that this is just a phasor plot in the way I measure. I don't say anything about the phasor plot, but I have a series of points here. I circle those points with the cursor, and I color the image. And of course, I get the cell, because that cell, and that has to be the GFP. And then if I move the cursor in the other position, the other extreme, I measure simply the surrounding collagen, which has a little fluorescence, and you can see that I can obtain the two extreme, 1 and 2, directly from the image. I don't need to know what they are. And then you say, what are the points, for example, along this line? Well, along the line will be points which have a little bit of fluorescence from EGFP and a little bit of fluorescence from the collagen. And you can see that those have to be in the border of the cell, where, of course, things add in order to do a linear combination, whatever. And then you will say, well, you see, I did the analysis without knowing anything about the system. And this is really, and I have, well, in here I have at least 100,000 points. And I did the analysis without knowing anything. I don't need to be an expert because the measurements are giving me the answer. But let us see more in detail. If I look more in detail, I see that actually there is another linear combination here. And I will show with different colors. So that linear combination, this is the point which gives me the cell. This is the point which will meet the collagen. But if I look at the collagen only, it has another component, which, so this is the collagen only, that will mix with another component, which is simply noise or autofluorescence of the sample. And so I have a series with 1, 2, 3 components, and I analyze for 3 components, and I can tell every value of the components must be along the line, because those of the line are given by the measurement. And so directly from the measurement, I get that there are 3 components. That gives us a general rules that if I have 2 components here and here, and doesn't need to be on the universal circle, simply the linear combination will be along the line, this red line. But if I have another component, every point of this component will linear linear combined with the points along the component. So if I have 3 components, the points must be in a triangle. And that is a representation that you don't have in the time domain. So here you look at the phasor plot and you simply say exactly how many components I have and where are those components. And this is the enormous advantage. So in the original work with Michel Digman and the other people in Spain, we have sample, for example, of YFP, which is here, CFP, which is here, EGFP is there, and you can see fluorescent protein to be different in all parts. We have the autofluorescence of the fibronectin and the autofluorescence of the cell. So, and this is something has to do with FRET. So you can see immediately that although things can be very complicated, I can distinguish 1, 2, 3 protein, for example, and the fibronectin and the autofluorescence, and I can have all the linear combination of one with the other. And this is something that you cannot do directly with the time domain. And this is very important. And also, it's very important that you see that the points are relatively in a narrow place in the phasor plot. Okay, so how can we distinguish between multi-exponential from FRET? Okay, so if it's multi-exponential and they have 2 points, which I draw on purpose out of the universal circle, the combination of those 2 phasors will give an experimental point that must be in the line between P1 and P2. If I have a quenching, which means FRET, for example, the FRET will change the lifetime. So the point cannot be along the line of P1 and P2, but it has to be outside. It has to be in that side because the lifetime will decrease due to FRET, and then we'll have a different trajectory. And the different trajectories that I have, of course, are difficult to calculate, but I can have a computer to calculate automatically that curve and superimpose the curve in the graph. So for that reason, we have in the program different calculator, we call calculator, to calculate, for example, the combination of 2 components. And things have to do with FRET. And this is also very, very useful. For example, the donor will be quenched, so will move in that direction, and the acceptor will be excited with a delay, and then will go out of the universal circle. So, and that is the formula I showed you at the beginning, how to calculate. So let us start to see how the FRET calculator works. So if the donor is exactly in the universal circle, so it's a single component, then the quenching trajectory, as you quench due to the presence of the acceptor, is still a single exponential and will move along that line. And eventually will go to 1 when it's completely quenched. But if the donor is not a single exponential, will be in a different point, for example, inside the universal circle, still we have a trajectory that will go to 1, this point, because that is fully quenched. So we will have a trajectory. But we will see that actually the point 1 is never reached, because as we quench more and more, autofluorescence will start to come out. And so if the autofluorescence, for example, is in this point, that will be the trajectory. The trajectory will be to end up to 1, but because there is autofluorescence, it starts to bend when there is very little fluorescence due to the molecule that we are we're working in this part. So for example, now you can obtain, if you have only autofluorescence and only donor, you know how much you are, how much autofluorescence is to respect to the donor, because that will be a linear combination. But this is the FRET calculator. The FRET calculator, start from whatever the donor. This is experimental point, and then we'll show a line. In that line, that point which contains the experimental point will give you directly the FRET efficiency. And you see there are 3 significant digits. So what I say is that the phasor does not, it is a very quantitative, you can do quantitative analysis. It's not just a representation to do exponential analysis. But for example, in the case of FRET, can give you the. So let us start with a thing which has importance only because that was done originally. So I have my collaborators that were trying to analyze these cells. So if you consider point A, you will get multi-exponential. At point B, you will get multi-exponential. C, you will get multi-exponential, and D, multi-exponential. And then they know that the color here is given by a U-power receptor that has EGFP, and then there is another U-power receptor which has a monomeric red fluorescent protein. And then this is the counter pixel. So to give you an idea, I say that you have about 500 pixels. Well, you have points which more, those are the larger point, but you see, this is the histogram of the intensity. So you don't have a 10,000 count. You have, let us say, below 1,000. Most of the thing is 1,000. So this is important because now we want to do all the analysis of the FRET and so on. And by the way, they knew from other kind of measurements that the FRET will only occur at the junction between cells. So let us see what we do. So first of all, we do the usual analysis that is done with the time-correlated single photon counting, the background figure 830 cell. And then you can see, first of all, that you get a distribution. And you will say, well, this is no FRET and this is FRET because it's decreased. But this is not true, because if you see where they come from, so the longer lifetime comes from this region, and this is the lifetime, and then the shorter lifetime comes from the other region. But this is simply that these cells are expressing more of the protein, this is expressing less of the protein. And you can see the separation of the cell very well, and you don't see any signal coming from the boundary between cells where it's supposed to be FRET. So this is clearly, they were stopping there and they say, well, either everything we did in biology before is wrong, or FLIM doesn't work. Well, I say, let us start to see what will happen. So the EGFP is here, and this is the autofluorescence. So, and I draw a green line that goes from the GFP to the autofluorescence. So I say every point which is along that line must be a linear combination of EGFP and the autofluorescence. So I circle in white this part and I paint one cell. So that is the cell that has more GFP than autofluorescence. Then I move the cursor down along the line of linear combination and I see the other cells. And then eventually I need to figure it out where is the FRET. Well, the FRET cannot be along the green line, the line of linear combination, but must be in the right side here because have to be a shorter lifetime. So I have the computer to draw all those lines. And then if you look carefully, there are points there of the phasor plot, which are in that region. And those point paints the junction between cells as it's supposed to be. So you see that I have done this analysis, never did a fit, and I realized that this plot is not correct. So this plot is a simple linear combination, but there is FRET, and the FRET is where it's expecting to occur at the junction between cells. But you have to look for that in that region. And I can see that it will be very difficult to do a fit in which that is multi-exponential, that is multi-exponential. Every point is multi-exponential, and figure it out that there is a region of the phasor plot that is not, is just FRET. And you can see that in this analysis, I never did a fit. I never did any complicated mathematics, but just showed the points, and I use a reasoning to place the cursor. So I will not talk about that because I don't have time, but I want to talk about something else that has to do with I don't have that in this presentation. So I will talk about the use of biosensor, as I said in the title. And the biosensor will be something that is the donor, that is the autofluorescence, and it has to be in that side of the, because in a biosensor, for example, let's see what they have the biosensor. The biosensor have various states in which the CFP can transfer energy to the YFP when RAC1 or CDC42 is present, and then that will be in the presence of FRET. And I want to understand how this stack works. So I have a donor only. So the donor only has, well, is uniform, relatively uniform, and that is the CFP, which has a lifetime of about 2.7 nanoseconds. So then I have to determine the background, and the background is in this point. So, so you can see that simply I have cell not expressing the biosensor, and that will give me the background. So now how I determine the FRET? Well, this is the experimental point. So the only thing I have to do is that this is the line of linear combination with the autofluorescence. This is the experimental point, and I move along this line. So moving along this line, in different positions of the line, I get the different activation of the sense. And for example, at 28% FRET efficiency, I get that part of the cell. At 50% FRET efficiency, I get the other part of the cell, and so on. And I can determine with very high precision the FRET efficiency in every point of this image. So, and this is the final plot, and I just moving along that line, I will get all the FRET efficiency, and this is the point of 0.472, and as you can see. So, and then we did for not active biosensors, so biosensor cannot FRET and so on. And in that case, we see that we can use biosensor to sense almost every molecule we want in the cell using proper biosensor. And this is actually what we did with Liz Hinde, who is now in Australia, and we determined the behavior of many biosensors. And in every case, we use this approach in order to calculate the FRET efficiency. So, so let us see if I can conclude this part. So many of the obstacles in the FLIM analysis can be removed, and the other important thing, the accuracy of extent determination is improved in the phasor plot, because the phasor plot has the property that you can do spatial filtering, and the spatial filtering is really what gives you a very well-packed phasor plot. The speed of the analysis is reduced to almost instantaneous because you just move cursor, and so it is really instantaneous. The analysis is global because I analyzed the entire image, and I can see if there is a region of points which are different lifetime immediately that I cannot determine with other methods. The interpretation of the FLIM experiment is straightforward. So it is essentially everybody can do. You don't need to be an expert because there is not a fit to be done. So you don't have initial values and so on. The phasor method can be applied to all modes of data acquisition, frequency domain or time domain, as I show in the original slide. And ion concentration, for example, can be calculated, and we did several work about that. So in conclusion, by representing molecular species rather than the sum of exponential decay, the phasor approach reduces the problem of fitting exponential. This is very important, and I didn't say specifically a lot about that, that really you only analyze for molecular species. And of course, this is important because the analysis in terms of molecular species is what you want to do from the very beginning. The analysis of the trajectory in the phasor plot provides a quantitative, this is very important, resolution of processes such as linear combination of two components or the calculation of FRET efficiency. We don't need to be an expert in spectroscopy to resolve the molecular switches present and to calculate ion concentration. And if global analysis, I can analyze also many different cells simultaneously and to see how they behave and have a global fit of everything. And I think this global analysis is a radically different approach. And I think there will be presentation by Lorenzo Scipioni that will show that we can do at the same time lifetime and spectra. I think Suman Ranjit also will show application to other sample, biological sample and so on. So I will finish here.